So now we're going to move into the actual cadaver structures. We're going to get into the brain, which is the second part of this, this week's topic. I'm not going to talk about the lab guidelines or lab rules and protocols for the cadaver lab. I'm sure your instructor will talk to you about that before we get into the cadaver lab. But the next set of structures we're going to be able to appreciate on our actual cadavers, okay? So we'll start off with the brain, and we'll first talk about the coverings of the brain, which are called the meninges. So the meninges are these coverings that are actually layered over the brain. So we talked about the skull a whole lot. It's this dense, hard casing or protective shell for the brain. And the brain is really soft, jelly-like, squishy, delicate tissue. And so in order to have this very soft, delicate tissue inside this very hard skull, we have to have it wrapped very tightly in these meningeal layers. So the meningeal layers help to offer additional support to the brain. It helps to also create a space for fluid, which we call cerebrospinal fluid, to circulate around the brain so that the brain can be suspended in the skull and not necessarily sloshing around from side to side. It's actually suspended delicately in this fluid. And so these are some of the function of the meningeal layers that are around the brain and in between the skull and the brain tissue itself. The first one is called the dura mater. This means hard mother. This is the thickest or toughest layer. The dura mater has two modifications, which is where it slightly changes its shape and it has a different name in two areas. So the first modification is called the tentorium cerebelli. This is an area that separates one part of the brain, the cerebral cortex, from the cerebellum, which is beneath. So we've got this modification of the dura mater that separates two parts of the brain, the cortex on top from the cerebellum underneath. The second modification of the dura mater is called the falx cerebri, and that sits in between both hemispheres of the cerebral cortex. So if you imagine thinking about a brain, and I'm sure when we get into the lab and you're able to hold a brain, you'll appreciate this much better, but it has this invagination right down the center, and that is where the falx cerebri actually sits and separates both halves of those cerebral hemispheres, hence the term falx cerebri. So the other two layers of the meninges, the layers that are beneath the dura mater, is the arachnoid mater. This translates to spider-shaped mother. This is not as thick or as tough as a layer as the dura mater was. As we can see here, this is a much more network or meshwork type layer. This is the layer that has the cerebrospinal fluid beneath it, so in between the pia mater and the arachnoid mater, what we call the subarachnoid space. This is where we find the cerebrospinal fluid. But in the arachnoid mater itself, we have some delicate blood vessels and so on that are running in between this layer. So not as thick a layer as the dura mater. It's much more of a network of a layer. It's got blood vessels in it, and beneath that layer, we have the subarachnoid space where we have cerebrospinal fluid. And then the third layer, the deepest layer for going from superficial to deep, is called the pia mater, translating to tender mother. This is the most delicate layer of the three. So we progressively get from thick and functional and tough all the way down to the very delicate pia mater, which is really a continuous structure with the brain parenchyma and the spinal cord itself. So it's really hard, almost impossible to distinguish the brain from pia mater. Pia mater is understood as a layer that is actually covering the spinal cord and the brain, and they're pretty much inseparable. So those are our three meningeal layers. And again, in between each of those layers, we have spaces or potential spaces in the event that there's bleeding or any type of injury to the brain or the meninges. So we have the subarachnoid space that is between the arachnoid mater and the pia mater. And then we have the subdural space, which is actually above the arachnoid mater and underneath the dura mater. Let's move on now to speak about the parts of the brain. So again, when you hold your brain, you're going to see that it separates into two distinct regions, or two hemispheres, we call them. This part of the brain is called the cerebrum. This is the top part of the brain that kind of looks wrinkly. Then we have the brain stem. So the brain stem is the central portion that continues on to become the spinal cord. All of this is called the brain stem. This has some other parts that we'll talk about further down. And then the third part of the brain is the cerebellum. So the cerebellum is this posterior part of the brain here. So it's easily distinguishable from the cerebrum itself. And it's going to be responsible for some other unique functions that we will talk about. And so I want to take this opportunity to point out where the tentorium cerebelli lays. So it lays in between the cerebellum here and the cerebrum on top here. So here's where we find that modification of the dura, the tentorium cerebelli. And then in between this half section, or this hemisection, is where we'll find the falx cerebri, that second modification of the dura. So let's go through the various parts of the cerebrum. So the cerebrum, we mentioned that first general top wrinkly, so to speak, part of the brain. It's got 4 separate lobes, and it's really nice because the lobes are named according to the skull bone in that region. So if you learn the skull bones really well, then you should know the lobes as well. Each of the lobes are paired, however, so bear that in mind. So we have 2 frontal lobes, 2 parietal lobes, 2 temporal lobes, and 2 occipital lobes. The frontal lobes are going to be on top and in the front. The temporal lobes are going to be on the side. The parietal lobes are going to be the very top and in the center. And then the occipital lobes are going to be posterior most, right on top of the cerebellum. Okay, now again, looking at the cerebrum, we appreciate that it appears very wrinkly. And so we can distinguish the wrinkles of the cerebrum from the actual grooves that are created by these wrinkles. So the wrinkles themselves are called gyri, and the grooves that are created because of the wrinkling of the brain are called sulci, or sometimes called fissures. So there are 3 unique fissures or sulci that we're going to describe. The first one is called the longitudinal cerebral fissure. This is the central fissure that separates the left and right hemispheres of the brain. It goes right down the center. This is the exact area where the falx cerebri resides or lies. The second one is the central sulcus. And the central sulcus is this invagination in between the parietal lobe posteriorly and the frontal lobe anteriorly. So we've got the central sulcus that pretty much chops or divides the cerebrum into the anterior frontal lobe and the middle parietal lobes. The third groove or fissure is called the lateral fissure or the lateral sulcus, and that is an invagination that separates the temporal lobe from part of the parietal lobe and more so the frontal lobe. So this groove that distinguishes or separates the temporal lobe off to the side and the main frontal lobe and parietal lobe is called the lateral fissure or the lateral sulcus. Next, we'll talk about the gyri. So what are some of the unique names of those actual wrinkles of the brain? So the gyri, there are 2 unique gyri we're going to speak about, the postcentral gyrus, also called the somatosensory cortex, and the precentral gyrus, also called the motor cortex. Now, what's unique about these 2 regions of the brains, these 2 gyri specifically, is that the postcentral gyrus, which is the gyrus that is posterior to the central gyrus, right? If you remember this, to the central sulcus, excuse me. So if you remember that central invagination here, the gyrus that is immediately posterior to that invagination is called the somatosensory cortex. And that area of the brain is responsible for our sensation in various parts of the body. So if you look at this frontal view or this sectional view of that gyrus, we can see the different parts of the body that are being represented here in terms of the sensory component. So we can see the genitals, that's closer to the midline. This is the area of the brain that controls sensation to the genitals. We can see sensation to the hip. We can see sensation to the arm, the hand, etc., etc. Now, what we can see is that some parts of the body are a little bit larger. This homunculus is a visual representation of the proportion of which the sensory components are being reflected here in this part of the brain. So the hand appears really large, so does the mouth, because we have additional or extra sensory components that are responsible for the hand, for touch, as well as for the mouth, for things like taste and so on. So that's what the homunculus is, this visual representation of different parts of the body and how they are reflected in this part of the brain in terms of the sensory component. Now we can look at the same exact thing for the motor cortex. So the motor cortex is going to be the gyrus that is anterior to the central sulcus. It's also called the precentral gyrus for that reason. And this is what controls the motor to the various parts of the body. So this is also called the motor cortex. So again, we can think about the homunculus as representing different areas of the body, and then this specific gyrus, the precentral gyrus, is going to be the parts of the brain that are representing motor innervation or motor components to those various parts of the body. Next, we move on to the cerebellum. So the cerebellum is that posterior-most lobe of the brain, or part of the brain rather. It's going to be distinct from the actual cerebrum on top. It's also going to look very different from the cerebrum. So it's going to be slightly wrinkled in its appearance, but not as wrinkled as the sulci and gyri of the cerebrum. Some of the features that we see on the cerebellum are the folia. So the folia appear darker in color, and this is an area of gray matter. We'll talk about what gray matter is, I'm sure, in our lecture. And that can be distinguished from the arbor vitae, which is an area of white matter, okay? So white matter, because it's going to be myelinated and appear more white, here, contrasting against this gray matter, actually looks like leaves on a tree. And so we get the term arbor vitae, which literally translates to tree of life, because of the contrast between the white matter of the central part of the cerebellum and the gray matter or the folia of the outer parts of the cerebellum. The third structure of the cerebellum is called the vermis, and the vermis is the central connection between both hemispheres of the cerebellum. So just like we saw two distinct left and right hemispheres of the cerebrum, we also have distinct hemispheres of the cerebellum as well. And those are conjoined in the center by this worm-like structure that's called the vermis. So the anatomists that named the vermis were obviously thinking it looked like a worm, just kind of sticking out in the middle here, and that's where it got its name, meaning vermin or kind of worm-like, and that is the vermis. Moving right along, we'll talk about the brain stem now. So just to remind ourselves where the brain stem is in relation to the other parts. So we have the cerebrum on top, we have the cerebellum posteriorly, and then the central stalk-like structure where the brain becomes the spinal cord is called the brain stem. The parts of the brain stem are the pineal gland, so the tiny gland that's found posterior to the thalamus here. The pineal gland is responsible for things like our sleep-wake cycle. It's also going to have connection to the optic chiasm, which are nerves that are associated with our autonomic nervous system, and the release of melatonin and so on. Again, connecting to our sleep-wake cycles. Then we've got the quadrigeminal plate. The quadrigeminal plate, sometimes referred to as the tectal plate, is really a collection of two layers of structures. On top, we have the superior colliculi, which is responsible for processing of visual information. And then on the bottom, we have the inferior colliculi, which is responsible for processing of auditory information. So these structures together forms the quadrigeminal plate or the tectal plate. Next, we've got the pons. So the pons is this outward or bulge-like projection of the brainstem. It's going to be an important landmark as we talk about some of the cranial nerves and so on that, that are traveling in this region. It's also a part of the brainstem that is associated with our integrating centers. So is the medulla oblongata, things like our respiratory centers, our cardiovascular centers that are parts of our autonomic nervous system, integrating centers in our autonomic nervous system that help to control things like the rate of breathing, the rate of respiration, even when we're sleeping or we're not actually aware of things like that. So the brainstem is really a connection of our integrating centers and our autonomic nervous system. That's going to be the pons and the medulla oblongata.